All right, good morning, everyone. So, as promised yesterday, we will talk a bit about metabolite identification. Now that I hope all of you could work your way through the tutorial on how to quantify them differentially and figure out what is going on, we will talk a bit about the identification of metabolites, which is a bit more difficult than with peptides. And that's another reason why I always tend to suggest quantify first and identify later. Because usually you have to identify only a small subset of what you really observe, and you can spend a lot of time and effort on identifying things that you don't really care about. We will see also that identifying metabolites is a bit expensive if you want to do it right. So the first thing I want to talk about today are metabolite databases. So when we look at metabolites, they tend to have very diverse structures. In proteomics, things are easy. They're linear. We have 20 amino acids. And these are just some selected metabolite structures. And you can see that there's a large diversity. We also have different elements that are popping up that are not so frequent in, or not present in peptides. That's all like halogens. Where can we find those structures? With the sequences in proteomics, you know where to find them. They're in UniProt and any of the protein sequence databases. And similarly, we have databases for metabolomics as well, and they basically come in three different kinds that are relevant to what we're doing here. We have structural databases that fundamentally store the structure of the metabolites. We have pathway databases that relate how these metabolites are involved in a biological network, in enzymes, and we have spectrum databases that contain actual mass spec data for each of these metabolites. In contrast to sequencing data, many of these databases are commercial, so you can buy spectrum databases from mass spec vendors or from other sources. Only recently, the community has started to make an effort to make more of these resources available. So let's look at some of them. They are all similar in a way. HMDB, the Human Metabolome database, is a database of, as the name implies, human metabolites, rich in metadata, so you actually learn what the role of this metabolite in metabolism is to the extent it is known. But it does not contain any mass spectra. So this is where you can get additional information on your metabolite. What does it do? Does it make sense that I see this metabolite? And you can try to figure out how it relates to other metabolites. Looking at this database, you can just enter it by searching for a name. For example, glucose, and you see one of the issues that we have already here. In contrast to peptides where we have one sequence, searching by name is sort of an inexact science by definition. So if you search for glucose, you will find glucose as the first hit, but you will also find all sorts of other things, structures that are related to glucose, that contain glucose. And if you click on the right entry, then you get something like a record for this structure. Well, of course, the molecular structure. You get some hierarchical classification. So this is an organic compound that is a carbohydrate, a monosaccharide, and belongs to the mammalian metabolites. And then you have sort of some annotation on the chemistry, where it belongs, and then, of course, the monoisotopic molecular weight, the average molecular weight. And this here, who has ever seen something like that? 3. This is the so-called SMILES, and that is a string representation of a molecular structure. That's something that you might know from cheminformatics, if at all. That's how structures in large compound databases can be stored. It's not the perfect way to do it, but it's one way to do it. And actually, if you copy and paste this SMILE, you can also visualize the structure in KNIME. You just copy the SMILES to a table, convert that to a structure in the way I showed it yesterday in the workflow, and then you can directly see the structure in KNIME. You can also drill down a bit and search for a mass that you observed. If you observed a positive mode mass of accurate mass of 181 point something, you can enter the mass here in this search window and ask HMDB to give you all the metabolites that would actually hit this mass. And you can see this list can be reasonably long for any given mass accuracy. Of course, glucose is within this list, but it's also a nasty example. Why is it a nasty example? Because all these hexoses are, of course, isobaric, and they're isomers. They're just stereoisomers. So you get a full enumeration of all the monohexoses here. At the same time, you see also other things like aspirin can pop up, not exactly the same mass, but these lists can get several hundreds of entries long, depending also on the mass accuracy that you use. While HMDB fundamentally contains a spectra of mass metabolites, this data is not downloadable as bulk, but you can look at it and you can see whether what you see in the database actually fits to your spectrum, but that's your manual effort, and that makes it a bit awkward to do that in an automated workflow. But you can look at the spectrum, you see the masses annotated, nominal masses, and if that fits to what you see in your data set, then that might be a useful hint. You can also search for spectra. So if you have a peak list, you can copy the peak list. You need to define the parent mass, you need to define the instrument type, and then it will try to pull out a matching spectrum from the database. But again, something that is done manually. So if you really do quantify first, identify later, if you deal only with 10 of those guys, it's easily done manually, but we will see later how you can also do it on a larger scale in an automated fashion. Another large database is Metlin. Last time I checked, it had over 240,000 metabolites. Note that these are no longer just human metabolites, but there can be anything in there. Plants, for example, are very rich in secondary metabolites of interesting structures. And you can search in a similar fashion that we just did it for metabolites and for mass spectra. It's very similar, which is why I won't walk you through the forms. It's going to look different every year or so anyhow. So I would spend like a day just updating these screenshots, but you get the gist. It's always the same thing. You enter a name or an ID or an accurate mass or a spectrum, and then each of these websites will give you something back. Another interesting resource is MassBank, and MassBank is something we use as well, also in OpenMS. You can download all of MassBank, which makes it very convenient. There's an interface from R that allows you to also use MassBank from R. It contains lots of metadata, permits the search of metabolites via their mass spectra, so that's another helpful resource. Perhaps the most interesting development over the last years has been GNPS, Global Natural Product Social. It's a database where you can also upload your data, and it's trying to sort them by spectra, cluster them. And if it can't identify that, but it has seen that already 10 people have seen the same spectrum, then if one of these people identifies the spectrum and adds that identification, then all of them will be notified that one of their spectra has been identified, which is kind of cool. So it's sort of a community effort to increase the identification rates here. We also have a version of MassBank that has now its own interface. That's Mona, maintained by Oliver Fien's lab at UC Davis. MassBank of North America has a much nicer interface, also great data resource. You can go there, have a look at it. You can also go to the MassBank vendors. There's mzCloud that contains a bunch of spectra at very high quality. You can imagine Thermo Fisher does not only sell the instrument, they also sell the compounds. So they just decided to acquire mass spectra of all of them. again, not a very helpful resource if you want to do any automation, because they're very awkward interfaces to work with this. The next set of resources are databases for pathways. Most of you will have heard of CAG, of the Kyoto Encyclopedia of Genes and Genomes. Here we can find both metabolite structures. Basically, for everything that is in their pathway maps, they also have the structure of the metabolites. You can download them. But more importantly, they actually have information on how these metabolites are connected in metabolic networks, and that can be helpful. Oftentimes, when you see the results, you also want to understand why is this metabolite upregulated or downregulated. Then it helps to go into the pathway and understand where it comes from and where it goes, in order to get some idea of how that would also relate to other data. We're doing more and more multi-omics, so relating that to proteomics or genomics data might be helpful to make a more comprehensive, more conclusive story. Then there's human psych that contains also a very comprehensive set of metabolic pathways, as the name implies, for humans, but there's also an eco-psych for E. coli, so you should check these databases out as well. They have a much, as far as I'm concerned, much nicer visualization of the pathways, much more interactive than CAG does it. And then often overlooked but very important for metabolomics is PubChem. PubChem contains millions, tens of millions of structures by now, and you can search PubChem by substructures, you can search it by accurate mass as well, you can search it by name. A tremendous resource, because fundamentally, once someone came up with the structure and wrote an article about it, you will eventually find it in PubChem. And there are other related databases, for example, ChemSpider, that contain similar types of information. And these database sizes are growing steadily. So again, I apologize that I don't update them like every half a year, but you get the idea. The number of metabolites stored in there is growing still very rapidly. That has to do with an increased interest in the field of metabolomics, but also with the availability of just more data. People are looking at that and trying to understand it. And you can see that there are many compounds. This year, 63 million. That's from a number from 2015 from PubChem. That number has gone up by I think another 10 or 15 million by now. So the space of compounds that we can look at is vast. The question is now, how can we find data in there? And one essential part is that we also start sharing data. I mentioned that at GNPS already. If we all put this data together, then every lab that has 500 spectra of their sort of pet compounds in there and thinks they're immensely valuable, misses that if everyone would share them, they wouldn't have 500, but they would have 50,000 or 100,000. And that is why depositing the data in the same way we also need to do that in proteomics is essential for metabolomics, perhaps more important here. And I would strongly encourage you, if you measure data, if you generate data, convince yourself, convince your PI, convince your funding agency. It needs to be deposited so that other people can also reproduce what you're doing. For metabolomics, Metabolites is one of the repositories that I can recommend. It stores all the raw data, but it also stores the interpreted data from your study. So you can put in the identifications as well. And it makes it easy for you to reproduce other people's work, because if you publish a metabolomics papers and you put in the metabolites ID and say, our data set is available at metabolites ID MTBS 234, then everyone knows what they're talking about, and you don't have to write awkward emails. I would like to get access to the raw data, and then you get an email from PI, very nice and polite. I'll talk to the PhD student who did it, who has long moved on, and you never hear back. You know how that goes, and it's just not good science on all sides. So please deposit the data. It's actually fairly straightforward. Once you have then this a metabolite's ID, like MTBL is 217 or so, you can also see the metadata. So in this case, you see these were 78 cases of hepatocellular carcinoma measured on a UPLC QTOF. And then you can drill in and you can actually get the individual files. You have some data on the runs, which file is actually what, which are the diseased, which are the control patients. That is also essential in many cases. And it just makes it much simpler to then download all the data and reprocess it. And if you have a workflow, you can reanalyze other people's data within a couple of minutes. And that makes it very convenient to also ensure that you are on par with what other people are doing and makes it easier to publish things. All right. But now back to my core topic. Knowing what I just told you, where you find the data, that's one thing, but identifying metabolites based on that, that's a different story. So we will talk about the distribution of metabolite masses, because that's a surprisingly interesting topic and perhaps unexpected to some of you, how they're distributed. And we will then look at two different methods, matching of MS-MS spectra against databases and accurate mass search. And then I will talk a bit more about database searches, something that I added very recently. So let's talk about identification algorithms. You can identify by accurate mass, of course. We've just seen that. You put in the mass and you get a list of candidates. So some people say this is not identification. Yeah, because you don't assign an identity, you just assign a pool of potential structures. But let's face it, if that is true, then there's also no identification of proteins and peptides in proteomics. So to me, identification is not a binary thing. Identification is a gradual process where you narrow down until you really have this one compound, figure out what this one compound is. And by narrowing down, I mean, it can be as easy as figuring out what compound class it is. Is it a carbohydrate or is it a lipid? That might already be enough for some applications, to be honest. And as you work your way down, this list of candidates hopefully gets narrower and narrower until you end up ideally with one compound that you're pretty sure about. But ultimately, what would you need to do to prove that this is the compound? I've often seen that from editors or reviewers. You have no proof that this identification is correct. So what would each of you accept as proof that this compound is correctly identified? NMR, yeah, so an orthogonal method that is independent, that's certainly a good idea, but it can be very hard to actually get an NMR of the pure compound because you would need to get it out of your mixture first. So that's not trivial. Other ideas? A synthetic standard. And what would you do with that standard? What would you do with this synthetic standard? Yeah. We've done that in one case. We got the spectrum. The spectrum was correct. We thought we had identified it. So we spiked it in the original sample, and the peak showed up at a completely different retention time. So, you know, these things can happen. Most reviewer will be happy if you show them the spectrum of your reference compound and say, you look, this is the exact same spectrum, but you have to run it on the same instrument as well, under the same conditions. That can cause issues as well. And as I said, it does not mean that it's really the same compound, because these metabolite spectra can also be very information poor. There are spectra where you got 5 peaks, and that's it. And there are actually many ways, there are sometimes several structures that can lead to basically or virtually identical spectra. So there are different degrees of evidence. And ideally, you would argue, I know that the retention time is correct. I know that the parent mass is correct. The spectrum looks more or less the same. I've validated it through a spike in. It actually pops up at the same position, increases the height. I've done NMR. I'm a true believer in this identification. But oftentimes you don't go this full length. At some point you draw the line and say, well, I'm reasonably confident these 20 compounds, there are going to be this, or this. And if you then really want to get a patent on the biomarkers, then you would probably go the full length and really make sure that you have found everything. But for the publication, it might be enough to just have a decent identification list. All right. There are different ways to compare to generate these candidate lists, and the easiest way is through accurate mass, but you can go via spectra as well, and there are fundamentally two ways to do that. You can compare experimental spectra to experimental spectra, or in a similar way that we did database search, you can compare experimental spectra to theoretical spectra, or as we are going to see in a minute, so-called fragmentation trees. So let's start by looking at accurate mass search. So masses are not distributed evenly, and metabolites or metabolite masses are not distributed evenly. So when you look at this table, it seems to be very plausible at first. Between 50 and 51 Dalton, so these are all 1 Dalton bins. 50, 100, 150, 200. So in steps of 50 Dalton, I just counted the number of PubChem hits that we have in this mass range. So the first thing that is pretty obvious is for low mass ranges, you have very few hits, because there are only so many ways in which you can combine a handful of atoms to give a chemically meaningful structure. But as you increase the size, the sheer number of combinatorial options in which you can puzzle these atoms together explodes. And here in the same mass range at 400 Dalton, you have already 130,000 possible compounds in PubChem. Now it gets a bit more interesting when you zoom in. So here on the right-hand side, we see the mass range magnified from 100 to 101 Dalton. So same mass range. These are the 2,180 compounds that were in PubChem back then. And you see that they are not spread out through this mass window evenly. But the vast majority is actually between 100.1 and 100.2 Dalton. Why? What's wrong with 100.6 to 100.7 Dalton? Why does nobody want to be there? Well, what's the monoisotopic mass of hydrogen, carbon, oxygen? Roughly 12, 1, 16, 14. So what is happening here is that the atom masses are discrete. And if you want to achieve a certain mass, then you need to find the elements that actually add up to this mass. And if they're almost all integral masses, then it's very hard to get numbers that are far off. So what would be your guess if you look into the bin at 300 Dalton? Would that still be the bin here between 300.1 and 300.2 that would be most occupied? Yeah. So it shifts as you go higher in mass, because these residual masses, basically everything after the decimal point, will also multiply and add up. So when you look at the mass distribution, at a bird's eye view, when you look at the whole mass distribution, this is what it looks like. The number of compounds grows exponentially with the mass. But if you look into the fine structure, you see that's actually this wiggling going on. So it's a very lumpy structure. Some numbers are very unlikely. And if you have an accurate mass that is in that region, then you actually have a small compound candidate list. But if you had hit something that is sort of here at the peak of this distribution, then you end up with literally thousands of candidates. And that makes it a bit unpredictable. Well, of course you can predict it, but just by knowing that you're in the range of 180, you can't really say how many candidates you would get. When you look at these isomers and look at where they come from, it becomes evident why you can have so many different compounds. These are some candidates for 117.0790, and that corresponds to C5H11NO2. And what you see here are all, or a subset of all possible ways in which you can recombine that. Now, if you have done organic chemistry, it's pretty clear to you what's going on. You have all these functional groups, and you can just rearrange them on the scaffold in any way you like. You move the methyl group left or right by 2 positions, you get a new structure. And oftentimes it's actually hard to distinguish these structures because they can have and will have very similar fragment spectra. When we look at the overall distribution of metabolites, note that these were organic molecules. These were not necessarily metabolites. These are all of organic chemistry. That's what's in PubChem. When we look at metabolites or known metabolites, compounds that have been annotated as metabolites, I should be a bit careful because we really don't know what the metabolome is. If we look at these known masses from databases, then this is what it looks like. The distribution of these masses tends to peak around 370 Dalton, or the average is 376. And you can see that there are a few metabolites below, say, 150, and there are few metabolites that are heavier than 500, 600 Dalton. That is to be expected as well, and to some degree that might already be a bias because we've only been looking in this mass range. Of course, there are some metabolites that are quite a bit heavier. So when we try to do a search in these databases, like with Matlin, there are options based on accurate mass and on tandem spectra. With Matlin, if you do such a search, what would it do? Well, all the input data you can already gather from this form. It would ask for peaks, and it would ask for a max of 30 peaks. So you manually paste the masses in there. You have to decide whether this is positive or negative mode. Why is that important? Or what is positive and negative mode anyhow? So in positive mode, it's just the voltage that you apply at the ion source. Positive mode, you generate positive ions. In negative mode, you generate negative ions. Some of these metabolites will generate ions much more easily in negative mode, carboxylic acids, for example. It's pretty evident that in order to make them fly, adding a proton is not going to help. On the other hand, if you have amines, for example, then adding the proton actually might help. So you see different types of metabolites, and our search engine needs to generate these ions because in the database, we actually don't have the masses that we're looking for. We have the structures, and the organic structures are typically uncharged. So if you select negative or positive mode, there are different ways in which they can be ionized, for example, by adding a proton or by adding an electron, and that has an impact on the mass. And then you search them. So what does it do under the hood? Fundamentally, this image looks very similar to what we saw in proteomics because it is. It's just spectral library search. We have an experimental spectrum that we turn into a list of peaks. We have a spectrum database, for example, mass bank that we downloaded. So we have library spectra. These are also experimental spectra, and hopefully from the same or similar instrument. Why would the mass spectra look different if they're from a different instrument? Yeah, different detectors, there might be different resolution, but there even might be different ways to collide the spectra. There might be different ways in which the ions are actually trapped and then used downstream. So you will see differences in these spectra, and they are very subtle, and oftentimes we don't really know why they're different, to be honest. We just see that the characteristics of this fragmentation will be different under these conditions. So if you have that information, and if you have spectra from a similar or equivalent instrument, then we compare again these lists and we get a hit list. Again, the danger here is that you will always get one hit, and you will need to have some idea of deciding on when you are going to believe that. Spectral library search is very sensitive to these parameters. If you search against the wrong library, you tend to get wrong identifications. It is not that easy to decide when you can really trust it. But there's one fundamental flaw. With peptides, if you really want to get a reference spectrum, you can just synthesize the peptide. You order it. You order it in China, a couple of days later, $50, you got your peptide. Very straightforward. Synthesizing arbitrary compounds, that is not so easy. Natural product chemistry, if some of you have worked in that, you can spend 3 or 4 PhD students on just getting a partial synthesis of something going. So you can't just buy that stuff. So for many of these compounds, we don't have a reference compound that you could buy. Nobody has it. And that makes it hard to actually get these experimental spectra. Another problem is that the spectra are often less information rich. Fragmentation can be very complex with metabolites, but why do peptides fragment so easily? Because they have this comparatively weak peptide bond. That's why they give you information-rich spectra. But you can imagine if you have a large annihilated ring system, that is not that easy to break apart. So what you actually end up with is a spectrum that contains only a handful of peaks. And then it becomes hard to figure out which of these different isomers it is. One way to then get additional hits if you don't have an experimental spectrum that you can match against is going for accurate mass search, because accurate mass search is actually the easiest way in which you can match structures in databases to mass spectrometric data. Again, a rather straightforward approach. You have an experimental spectrum. All you need now is the M over Z and the charge. And then you go to a Metabolite database, you take out all the structures, you generate now all the predicted exact masses for the charged ions. So you add a proton, for example, depending on the chemistry, and you add adducts. So this is usually done by having an adduct list, for example, ammonium, sodium, acetonitrile. You can just define that, and that will depend, again, on your experimental conditions. It's pretty evident. If you don't have acetonitrile in your mobile phase, you won't see the adduct. So you have to have some insight on the experiment to generate this list. And then the search is really simple. It just looks whether the mass that you observe is within a certain tolerance delta. And if so, it just reports this hit list. Note that this hit list is usually not ranked. You can rank it by the delta, by how much does the mass deviate from your tolerance, but that is by no means a guarantee. So accurate mass search is a way to give you candidates if your spectral library search fails or doesn't yield anything interesting. Adding these counter ions, these adducts, solvent adducts, making the ionization happen is relatively straightforward, but it's something that you need to control. If you sort of miss that this adduct list is there and you get it wrong, then you also start getting lots of candidates that are fundamentally impossible to get in your experimental setup. If you try that with Metlin, then again, we would input just the mass. We would input the tolerance. Where do you get this tolerance from? Yeah, that is what you would know about your instrument, hopefully. And what happens if you make this tolerance too narrow? well, then you will just miss candidates. So this value should be realistic. What happens if you make it too wide, if you're too cautious about it? Then you will end up with thousands of candidates that you can't do anything with. So a good thing here is also to recalibrate your data. Oftentimes you can just reduce the tolerance by a factor of 2 if you do proper recalibration of your spectra, and that gives you a much more informative, much more concise candidate list. So guessing this value is probably not a good idea. So you should be careful, should think about what you really observe, should look at your variation between experiments, and should think about recalibrating the spectra. And then you get such a list of candidates you can drill in, and then you have to go and validate them and see whether that makes sense in your biological context. Sometimes you can exclude them if you're pretty sure that there can't be any caffeine in your sample, then that's okay. But if you're working with urine, you can be pretty sure that you would find it there. It would make sense. So it depends on your context again. Limiting factors are usually the instrument accuracy. You can't really do that if you don't have enough mass accuracy. The other issue is database completeness. We can only find things that are in the databases. Now, if you're working in, say, secondary plant metabolites of things that haven't been studied before, then the odds that you find what you're looking for in this database are pretty slim. And now the problem will be that you still get candidates. Most of them will be metabolites from animals, most likely. So you have to be cautious about that. It's not that easy to do it. The other problem here is you would like to have some sort of FDR control, and that has been a big issue in metabolomics for a long time. There are now some ideas on how to do this. I won't go into the details, but there's a Nature Methods paper that came out in 2017 that does it for accurate mass search. The problem again is this assumes that we have an idea of what the space of these metabolites is. And if we get this wrong, then also the FDR estimates are off by a bit. So how would we do that in OpenMS? I've shown that already, but now you have an idea of what's going on under the hood. This was the identification workflow that we discussed yesterday. So we start with the input files, and we have here an accurate mass search that is hidden in these subnodes. And we have here the spectral library search that just takes an external library, tries to find the hits. And this joiner node, again, brings it all into one table. Here we construct the structural drawings of the compounds. And then the interactive table actually gives us a bunch of candidates for different retention times, for different mass to charge ratios. And you can have many of these candidates for each feature. So this was basically all we could do until 3, 4, 5 years ago. There had been the idea in literature that you could also try to do something very similar to database search in proteomics. The idea is fundamentally the same. We would need to have a theoretical model of fragmentation, and people have tried to predict the fragmentation of metabolites for a long time, but it's not as easy as it is for peptides. If you can just predict B and Y ion series, then you're good to go in proteomics, and there's no equivalent in metabolomics. The closest, or the idea that works best, are so-called fragmentation trees. And note that this is not the same as a spectrum. For computer scientists, fragmentation trees are directed acyclic graphs that relate a molecular structure to potential fragment mass. That sounds a bit complicated, but it's basically giving us the relationship between different masses. It says this fragment mass can arise from this fragment. And fragments can break apart as well. That's how fragmentation works. You can break them down into smaller bits and pieces, and that is what this fragmentation tree captures. The idea would be, if I have the structure, if somebody drew the structure on a board, I could just go to the board and try to wipe out any of the covalent bonds. And that would make for 2 fragments most of the time. There's one exception. When doesn't breaking a bond give me 2 fragments? Yeah, in cycles. Yeah, if you break a bond in the cycle, you get still 1 fragment. So there's some issues here. And it's also not easy to predict which of these bonds are going to break. You can try to predict that with quantum chemistry. You can imagine that you can spend a couple months just doing that for 1 compound, so that is not a very scalable solution. We actually spent 2 years on that. I can tell you that is a fun exercise, but it doesn't scale that great. So instead, you can try to learn these things. And you can try to enumerate all of them. And the 2 things are kind of contradictory, but I'll show you how that you can do both. If you enumerate all of the possible fragmentations and then learn which of them are likely, then you end up with fragmentation trees that are helpful for the identification. So there are different types of fragmentation trees that people have used that are in the literature. One way is to decompose the molecular structures in the way I just described it. You break a bond, you get 2 fragments, you know the structure of these, and if you know the structure, of course, you know the mass of these guys. The other is, if I don't know the structure, I can still take the molecular formula. If I have a molecular formula of C2H2, then there are only so many ways in which I can break that apart. And you can start enumerating all possible compositions. These are so-called compositional fragmentation trees. The first method then requires a structural database. The second approach relies only on sufficiently accurate masses. And with high-resolution mass spectrometry, you can actually determine the molecular formula based on the feature mass. We haven't talked about that, but it's feasible. It's actually reasonably straightforward. So we first discussed these structural fragmentation trees. That was suggested in 2006, I think, for the first time. And you start with a structure like alanine here. And you look at different ways in which you can decompose this structure. By breaking this bond, you would get this guy. And by breaking this bond and this bond, you would get this guy. From a computer science point of view, what we construct is a graph where nodes correspond to structures or fragments thereof, and they're connected by such an edge or an arrow if you can generate this fragment from the other fragment. That's all there is to it. So you start here you start breaking things off this molecule at every bond, basically, and what you get is a subfragment where this bond has been removed. There are some issues with cycles, as we said, with rings. But you can enumerate them, and that is something that computer scientists are good at. You just take this graph and you enumerate the hell out of it and see what happens. Source molecules can often be fragmented in a single step. That gives you tandem spectra, or you can also do that iteratively until no further fragments can be found. That would be ms to the n, ms3, for example, where you say, let's fragment this guy. You isolate that again. You can do that in a mass spec if you really care about the structure. And that contains useful information because oftentimes if this peak, if there are only like 4 peaks in there, you would like to know what these fragments are that are apparently stable, and you can fragment them and get additional structural information using ms to the n. So what people did with these structural fragmentation trees was they tried to come up with the best explanation for a tandem spectrum. Because if you know the structure, if you know the tandem spectrum, you can just fragment the structure until you cannot break it apart further. And then you look at all the leaves in the tree, all the nodes, the final nodes, and also the inner nodes, because they all correspond to fragments, and you try to find which peak would correspond to which part of this tree. And ideally, you would be able to find the most likely fragmentation pattern this way, because when you look at the experimental data and see which peaks occur, you can also see in your fragmentation trees which of these edges are occurring frequently. And that is comparatively easy to do. So we could use that for identification if we're saying we have a spectrum that has distinct peak masses, and we find what's called the lightest connected subtree of the fragmentation tree that explains all the masses. What is a lighted connected subtree? Well, connected means that you can go from the root to all the leaves. It doesn't fall apart. The lightest is that you take only the parts that you really care about. So you remove all the rest that is not explained by the spectrum or is not needed to get to these fragments. And in this way, you can figure out how this fragmentation actually occurred. It's just a guess at how it occurs, but it's an educated guess at how this fragmentation occurs. Heinonen et al. Suggested that the lightest fragmentation trees can be found using what's called a MILP, a mixed integer linear program. You can look into their paper if you're a real computer scientist. It's actually a fun read, but it's not that easily digested. It's just an optimization method that is able to deal with large combinatorial spaces. For the majority of metabolites, their method could already explain 80 to 90% of the fragments, which is kind of useful. It doesn't help us for identification yet, because all it does is it explains the spectrum that we have. It gives us an explanation, but we need to know which structure it actually is. You could say, well, let's generate for this structure all fragmentation trees, or for this spectrum, all fragmentation trees of all the compounds that would match the accurate mass. And that's exactly the direction they went. You can use that to get some hints at that, but it was a rather crude method at the time that was not very discriminative when it comes to identification. Another approach is compositional fragmentation trees. This Heinonen approach, structural fragmentation tree, is similar related to database search in proteomics, because you have to start with a known structure. And compositional fragmentation trees, in a way, are similar to what is called de novo sequencing in proteomics, where you don't have the sequence. All you know that there are amino acids in there, and you're trying to make up everything on the fly. So the idea is based on the concept of mass decomposition. As we've seen earlier in this lecture, the masses of a fragment of a molecule needs to correspond to a sum of existing fragments or existing atoms ultimately. So not all masses are possible. In turn, you can figure out which ways there are to create a certain mass. So it's kind of related to this money changing problem. If you want to give someone $5.25, there are numerous ways in which you can do it if you're given a certain set of coins and bills. And if you lack certain coins, then certain numbers become unachievable. If you don't have cent coins, then there's no way you can go to $5.26. And this way you can also decompose a given mass and say, what are all possible sets of coins that could have given me this value? These compositional fragmentation trees look similar to the structural fragmentation trees, but instead of having a true structure, we only have compositions. So we know that C22H24NO7+ can lose water and turn into C22H22NO6+. You see, you lose 2 hydrogens, you lose 1 oxygen. That's what we would call a neutral loss. And this way you can break the molecule apart by taking out plausible substructures. except that you don't really care about the substructure, but you look at sets of atoms that you can take out. So this type of compositional fragmentation trees does not really need to know what the structure of this guy is. It would still lead to reasonable fragment masses. And this works reasonably well also to explain complex spectra. Because each of these fragments you can then align back to one of these masses. And if you see which of these nodes are present in the spectrum, in this case here, you can see all potential fragmentation paths that this could take. Yes, please.
The previous slide you showed
Whether we predict the intensity? No, we don't. Fragmentation. Where do you see 4.8%? That's what you observe in the spectrum. Yeah, we're not predicting intensities yet.
okay.
Yeah.
So that's what you observe.
These compositional, all the fragmentation trees are always related back to an observed spectrum. They're trying to explain the spectrum. When you look into these spectra, you can, or in this compositional fragmentation trees, you can start interpreting your spectra. So they're actually an excellent tool to also manually validate these spectra. Because if you start with one of these fragments, like 120.1, that would be this node here, then you can see what are all possible fragments that can arise from that. And if you see these peaks here, you know that actually this here is the mother fragment of all these other peaks. And that can help you to puzzle together your structure, possibly with auxiliary information that you have from NMR, for example, and really try to ensure that the spectrum that you see is consistent with the structure that you have. At this point, we're doing this only manually. Very recently, fully automated approaches that are based on this concept of fragmentation trees and database search have arisen. And that's a paper that came out just a couple of weeks back in 2019. That's series 4. And series 4 actually does what you would like it to do. It tries to predict these intensities. It tries to predict these fragmentation trees. So you start with a spectrum on your own computer because computing these trees takes a while. You compute these trees, send them to a web server, and that has a structured database. There's a machine learning component in the background that tries to make these predictions. And they do that on a reasonably large compute cluster and push back the result of this computation. What you then get if you use series 4 is such a candidate list. You see the smiles of the compounds. You can look at the structures of these compounds. You can see the quality of these predictions, and you can walk your way through the different candidates spectrum by spectrum. The result is actually stunningly good, which is also why I think this paper was published a while back. So there is a blind structure prediction contest, CASME, Critical Assessment of Small Molecule Identification. And that has been held for a couple of years, where you release mass spectra of a compound, and you make sure that it's not in one of the databases, and you don't tell anybody what it is, and just ask several groups, leading groups in the field, tell us what you think this is. It's never going to be a large data set. Someone has to synthesize these compounds, make sure that it really is what it is. So these tend to be 100, 200 compounds that are put into the competition. And series 4 really performed very well in the last CASME. It could correctly identify 74% of the data set. So that's quite impressive. And in 84% of the cases, the right structure was in the top 3 candidates. So that is really working exceedingly well. A word of caution, however, these identification rates will vary with the database and with the difficulty of your molecules. There are just some molecules that are harder to predict than others. That won't surprise you. If your machine learning component has never seen a structure that is remotely related to that, if you have 5 boron atoms in it, then things will get tricky, and then you can expect much lower identification rates. But it just shows that we are actually moving to a point where small molecules can be identified with a database search, similar to proteomics. But there are still issues here, for example, the FDR estimation on these data sets. While there are methods to also do FDR estimation, it's not clear how reliable they are on large data sets. All right, with that, I'm at the end. I would say we get started with the hands-on part already. Unless you have any questions, then I'd be happy to take those as well.